Today we'll be covering unit or chapter 5 on metabolism and energy. So to start us off, let's talk about C.B. van Niel. He was born in 1897 in Haarlem, Netherlands, and then he's later going to die in 1985 in California. At first, he was interested in science at a young age. He enjoyed agricultural research and analytical chemistry growing up. He liked to play in the garden and to research different types of plants. He enrolled at Delft University and got a degree in 1922 and a doctorate in 1928 in chemical engineering. He became an assistant to a biochemist known as Albert Clervier. He was a professor of microbiology and a comparative biochemistry professor at Hopkins Marine Station of Stanford, and that was from 1929 to 1962. So that's how he made his way over to the United States. He then became a visiting professor also at the University of California in Santa Cruz from 1964 to 1968. So he did some work there. He gave his scientific contributions in photosynthetic chemistry and in comparative chemistry of microorganisms. So he's going to study how plants are able to take in light energy from the sun and to create that into glucose through photosynthesis. And then he's also going to look at different forms of photosynthesis in microorganisms that are able to perform this important energy process. He's first one to receive the American National Medal of Science in 1963. So he's going to be very in studying energy and metabolism. So let's go into what exactly is metabolism. It's going to be the sum of all chemical reactions that occur in a cell that are responsible for transforming energy. So any chemical reaction in the cell that is going to help you to break down and release energy or the form of ATP. Metabolic pathways are going to be reactions that occur within a cell in a sequential manner, requiring things such as enzymes. Remember, all of your enzymes are going to be proteins. There's two types of metabolic pathways. We have what's called catabolism. That's responsible for breaking down large complex molecules. And we have anabolism. That's going to be responsible for synthesizing or putting together new complex molecules. So let's think of you eating a piece of chicken, for example. In catabolism, you would break that piece of chicken down by its proteins, break those peptide bonds, into separate amino acids. Let's pretend it's Lego. So you break that Lego sandwich down into each one of those little building blocks. Each building block has a different size, different shape, maybe a different color that would resemble its amino acid. So there's 20 total amino acids in our foods. Think of 20 different sized Lego blocks that you could have, different sizes, maybe even different colors to give you the 20. So that sandwich was put together with different blocks. So you take those blocks, you break them down. So now you're able to use those Legos and rebuild anything that your body might need using those Legos, assuming that your body was built of Legos. So you take that's catabolism. You took the sandwich or the piece of chicken, and you broke it down into the separate Legos, and now you have those separate amino acids. Now you're going to take them and put them together. That is going to be called anabolism. So you're taking those separate Legos, and let's say you need to rebuild or repair skin damage. You scraped your knee, or you might have cut your arm. So it can take those proteins and help to rebuild the proteins that would be in the skin, or rebuild the proteins that might be in the muscle from working out that you could have tore or damaged, to rebuild it. And so catabolism breakdown, anabolism is to synthesize or rebuild. So you'll have independent reactions that are doing these things, but they can also occur simultaneously, producing energy and molecules that each can utilize during specific reactions. So as you're breaking these down, I forgot to mention, you're separating them so you have your different amino acids you can use, but you're also getting energy from breaking them down. So that's where you get your energy from the chicken as well. You're not just going to get your building blocks to build things or your amino acids, you're getting energy. The definition of energy is going to be defined as the capacity to do work. This is going to be used by cells for developing, growing things, repair, reproduction. All of those things are basically like the rebuilding or creating of something. So that would be your anabolism. These are going to be measured in calories. And everybody knows, especially if you're on a diet or you're trying to watch what you eat, you'll look at those calories to see how many are found in each meal or each serving that you're taking in. A calorie is actually going to be the amount of energy required to raise the temperature of 1 gram of water 1 degree Celsius. That is going to be the definition of a calorie, so know that. Kilocalories are going to be the most common unit for measuring the energy content of food and the heat output of organism. So as you're using this energy and you're breaking these molecules apart, there is always going to be some energy that's lost. And energy is always going to be lost in its most kind of what we consider to be the useless form, and that is going to be heat. So the energy that's going to be required to raise the temperature of 1 kilogram of water 1 degree Celsius is going to be your kilocalorie definition. It's the most common form. If you go to other countries, you'll see that the foods aren't measured in calories, they're measured in kilocalories. We call them calories here. So that is actually going to equal 1,000 calories because a kilo is going to be 1,000. Now the nutritional labels are going to be measured in kilocalories, like we said, but for the sake of our US metric or US measurement system, we just call it a calorie. So looking into this with our different types of macromolecules we've already discussed and studied in the previous units, 1 gram of carbohydrate is going to be made up of 4 kilocalories or 4 calories. 1 gram of protein is going to have 4 calories or kilocalories. But 1 gram of fat is going to have more than double that. It's going to have 9 kilocalories, or what we would consider here in the US a calorie. So coarse fats are going to have a lot more energy with them if you do not use that energy right away. It then gets stored as adipose or fat in your body. So there's different sources that energy can come from. So energy sources and flow is going to be very important. The main source of energy where most food products are going to originate from is going to be the sun. So we have solar energy. Geothermal energy is going to be another type of energy that can be used by organisms. That's energy that's provided by inorganic chemicals such as iron, sulfur, and ammonia sometimes. Energy flow is the energy flows from the sun. So we have our photoautotrophs. These are going to be the original organisms that are going to use sun that they capture. They're going to capture that energy and convert it over into a sugar energy that can be used. And they do that along with carbon dioxide and water. And they make those organic molecules that we will then use for cellular functions or for food or to run our bodies. So those are your photoautotrophs. Those are also going to be referred to as the primary organism there. After that, it's going to flow from them to others, the others that will eat it. So think of a cow eating grass. Grass is going to be your photoautotroph. Then you have the heterotroph or the omnivore that's going to come in, and they're going to come in and eat these things. So they eat the plant material and use it in their body. You can also have energy from inorganic chemicals, like we just said, and these are going to be referred to as the chemoautotrophs. These are going to be things such as bacteria and archaea that are able to obtain their energy from inorganic chemicals like iron, sulfur, and ammonia, and they're able to take those chemicals and make organic molecules out of them. So energy flow from these two primary sources, the photoautotrophs and the chemoautotrophs, are going to flow to another organism that will then eat them. And those organisms, like I said, with the cow as the example, are going to be your heterotrophs. So organisms that basically consume other organisms and rely on photoautotrophs and chemoautotrophs for all of their energy needs. So here you see the sun starting out as the primary energy source. It comes down to the producers, which are the plants, and also it can go over to the consumers when the consumers eat the producers. All of these things will decompose. When they decompose, things such as fungi, bacteria, and worms are then able to eat their material. So the decomposers can also be referred to as detrivores, and the decomposing matter can be referred to as detritus. Okay, different energy forms. We have what's known as potential energy. That is energy that is going to be stored, or energy that's available to do work based on their stationary position or based on their location. So stored energy, think of like a battery. If you have a ball sitting at the top of a hill that can roll down, it has potential energy. If you have a candy bar that is full of sugar, that is potential energy. Once you eat that, then your body can break that energy down and you can use it for food. So another one, you can have chemical energy, which is energy stored in chemical bonds of molecules, like I just said with the candy bar. That's a good example. Other examples could include water behind a dam, a teaspoon of sugar, that sugar's holding potential energy for your body, a snake that's poised to strike, he has potential energy, a rock sitting on top of a cliff, a baseball player ready to throw a ball, and a stretched rubber band that's ready to pop back, or a skier on top of a mountain. All of those are examples of potential energy. Next, you have your kinetic energy. That's energy that's being used to move an object. So this is the energy of movement. The first one, potential, was stored energy. Kinetic is actual energy, energy of movement. So various examples of kinetic energy are going to be things like thermal energy. They move molecules within matter. If you're not sure about that, think about if you're near a fireplace, or if you're near a heater and it's just emitting heat that's coming off. You can see little hairs moving from that, or maybe fuzz on something is moving because that heat energy is causing it to move. It's causing kind of like a slight breeze. Next one, you have solar. That's the movement of photons. Photons are going to be energy particles of light. You can have sound energy. That's the movement of the vibrational waves that are traveling through the air. And you can see this sometimes if you stick something on top of a speaker and you see it vibrate and move. So all of these are going to be examples of kinetic energy. You could have water flowing through the dam. That would be kinetic. Burning sugar, striking snake, a rock rolling down the hill, the baseball player throwing the ball and snapping a rubber band, or the skier skiing down the mountain. So you changed all of those from that potential energy over to kinetic energy. First one that you see is potential. The water's behind the dam. Then they open up some of the openings there, and then you see the water actually flowing. That's going to be your kinetic energy. This is going to lead us to the laws of thermodynamics. So this is going to be two laws that explain the flow and transformation of energy between and within organisms. So with the first law, the law of thermodynamics is going to state that it cannot be created or destroyed, but can only change form. So it can be converted from one form to another. An example of this is taking solar energy and converting it over into chemical energy through photosynthesis. So it captures that energy, takes it, and converts it over using water and carbon dioxide to make the energy that it's going to use, which is going to be glucose. So that's going to explain the flow and transformation of energy between and within organisms as far as the solar. Now the second type, you have second law of thermodynamics, which states that energy cannot be converted from one form to another without a loss of usable energy in the form of heat. And remember, this type of energy they consider to be the most, wasteful. We lose a lot of energy during a lot of processes through heat. So heat can never be converted back to another form of energy. And that's why they say that's the most kind of useless, usable energy. Lastly, we have entropy. That is going to be defined as a disorder in the universe. That's going to occur as the amount of heat is going to increase. It decreases the amount of energy that's able to do work. So as things heat up, machines become less efficient and you lead to entropy. This molecule here, you've probably heard of, ATP, but you might not know exactly what it is. So it stands for adenosine triphosphate. This is given its name because it's going to be made up of a sugar and three phosphates that are going to be hooked together. So you have adenine and the ribose together. That's your adenosine. And then the triphosphates will be three phosphates hooked together. So that's going to be the yellow portion of the tail there. So it's your main energy source within a cell and within living things. It's responsible for providing energy that's required for different types of chemical reactions. It has a nucleotide composed of the base adenine and your 5-carbon sugar that you see there. and then those 3 phosphates I was telling you about. Now, the primary source of energy, like we said, that's found in this ATP is going to be found between those phosphate groups. So I'll go ahead and get my pencil for a minute. So you have your 3 phosphate groups here that are bound together by a high energy bond, and you see it's pointing to one of those bonds. When you go in, if you break that bond, and it's a covalent bond, so if you break that bond, you're going to release a burst of energy. And that is going to be where the energy comes from to run your cellular processes. Once you break it, it's going to convert the ATP down to this right here, which is ADP, stands for adenosine diphosphate, di meaning 2. Of course, the one above, ATP, triphosphate, tri means 3. So you convert it over to ADP, and then you have this phosphate right here that you released. But the important thing was not just releasing that phosphate, it was the breaking of that bond right there that gave you that energy release that your body can use. So it's going to produce about 7.3 kilocalories of energy, and that's enough to supply sufficient energy for biological reactions to occur. So this, like we said, converts it to ADP. That's quickly resynthesized when energy is going to be released during glucose catabolism, so breaking down sugars, breaking down foods. So that ADP is kind of like a rechargeable battery. It goes back, picks up another phosphate, hooks it back on with a high energy bond, becomes ATP again, and is ready to be used for another reaction. So effective sources of energy for 3 reasons. With this ATP, it's constantly resynthesized, so it's constantly being rebuilt from ADP to ATP. It's covalently bonded, holding the phosphate group, so it's unstable and easy to break. And when it breaks, like I said, it releases that 7.3 kilocalories of energy, which really helps that cell perform whatever function it needs. And the energy is released when the covalent bond holding the last phosphate group is broken. It amounts to twice the amount of energy that's required for those reactions. It comes in and it really helps those reactions out a lot, gives them lots of energy. So it also is going to provide sufficient energy for cells to perform their main functions. So first function, chemical work. It's the energy is going to be used to synthesize important biological molecules. So it might need to make proteins. So it's going to take energy to do that. It might need to make a phospholipid bilayer. Everything inside that cell that it needs to make is going to take energy to do. It also will supply the cell with energy to transport substances across that plasma membrane, because remember, some things require actual energy at those proteins to push them out or bring them in. And it works as mechanical work, so it supplies the energy needed to perform mechanical processes like muscle contractions or propelling the cilia or flagella in the cell. Photoautotrophs are going to be organisms that include things like plants, algae, and cyanobacteria. And what they'll do is convert solar energy into chemical energy and synthesize organic molecules. So photosynthesis is going to be the process it does this in. So that involves the conversion of carbon dioxide, water, and light from the sun into glucose. And it's also going to release oxygen when it makes this glucose. So that's why we say that plants make most of the oxygen that we breathe. So it'll go from the equation that you see at the bottom. Hold on. So you'll take, give it a minute. All right, here we go. So you'll take your carbon dioxide, which is CO2. You'll take some water, which has to come in through the roots, and you'll take sun, so solar energy, which will come into the chloroplasts and be converted. And in that process, it produces, that's what that arrow means, glucose, which you need to know is C6H12O6, and breathable oxygen, which is O2. So those are your reactants. So reactants over here, reactants over here, messed up, products over here. So what you're putting together on the left side, what you're getting on the right side. So like I brought up, the capturing organelle is going to be the chloroplast. It's a double membrane organelle that's found within the mesophyll cells that are going to be located between the upper and lower epidermis of a leaf. Now, the upper epidermis of the leaf is going to contain a waxy substance called the cuticle. That's going to be very important because it's going to keep the plant leaf from drying out. So it kind of helps keep it from desiccation. Desiccation's drying out. The lower epidermis is going to contain the stomata. Those are going to be small pore openings. That's going to allow carbon dioxide to enter the leaf, and it's going to allow oxygen to exit the leaf. So those small holes that you see are going to be referred to as the stomata. So that's the hole that's inside. Now, what opens and closes the stomata are going to be two cells, one on each side. They are called guard cells. And so here's a guard cell, and then here's a guard cell on the other side. So they'll open up that hole if there's plenty of moisture, if it's especially nighttime, it needs to release oxygen and bring in carbon dioxide, things like that. But if it's a very dry day, it might close those stomata to keep that cell from drying out or that plant from drying out, and it would actually have a buildup of oxygen in there and not enough carbon dioxide. So it might have to open them up later in the evening once it's not as hot in order to have its gas exchange. All right, so one mesophyll can contain up to about 200 chloroplasts. Inside the chloroplast, you'll have what's called the stroma. That's the gelatinous fluid that contains DNA, ribosomes, and other various enzymes. So that's going to kind of be like the cytoplasm of this organelle. It's called the stroma. This is also going to be the location of the synthesis reactions. That's where the chemical energy is going to be used to synthesize sugars. The thylakoids are going to be interconnected membranous structures that are organized into stacks. These stacks are known as grana. So over to the side, you see each one of those flat pancakes. A stack of those pancakes is a grana. This is the location of the photo part of the reactions. So this is where the light energy is converted to chemical energy. So light energy is going to be caught at the thylakoids, and then it's going to be sent over to the stroma in order to make the sugars or make the energy. Chlorophyll that's found within the thylakoid membranes is going to be a pigment, and that's the pigment that's involved in capturing that solar energy. So it energizes the electrons prior to the reduction of carbon dioxide to a carbohydrate, and that carbohydrate is going to be glucose usually. So visible light is going to be an actual type of kinetic energy. It makes up a small portion of the electromagnetic radiation or the electromagnetic spectrum that we talk about lots of times that hits Earth's surface. So actually less than 1% of that helps to provide the energy. Or you might think about it as your Roy G. Biv. Red is going to be the largest of the wavelengths of light, but they move the slowest. As you go across that spectrum, by the time you get to blue, indigo, and violet, these are going to be the fastest moving waves that carry the most energy, but they will be the smallest waves. Now, in space, or if you're talking about light energy, since waves are really, really big, even though they have less energy, they move really big and really slow. That energy can usually travel pretty far. And so that's why lots of things that we see, some of the older things in the solar system are going to be red-based because the energy is still traveling to us. Now, the fastest things that are moving, the indigos and the violets, those types of things move with so much energy and they are so fast, but the wavelengths are small. And so they will fizzle out or not make it as far as those red wavelengths will. Right. So with this, the Roy G. Biv theory is going to be deduced through experiments that were performed by Sir Isaac Newton in 1672. So here's a chart that's showing you what I just described. So in this, you're going to see all the way over to the right-hand side is going to be your smaller waves. So these will be your radio waves. These are the largest waves, not small, but the largest waves that carry the least amount of energy but travel the furthest. So radio waves can travel very long distances. Next, you have coming in with your radio waves, you'll have AM and FM. AM will actually travel larger distances. It'll have larger waves, but less energy in it. As it cuts over or moves over to FM radio, that's what we tend to listen to the most when you think about radio. Those are going to be slightly smaller waves that don't travel as far. So you can have an AM radio channel and listen to it longer for longer distances as you're traveling away from where that radio station is than you can with an FM radio. Now we're getting a little bit faster, a little bit smaller waves coming in. We have the microwaves. And so we'll use those to do things like cook. And then coming in, you have what we refer to, and we've been talking about, we have our visible light waves right here. And so with the largest waves and the least amount of energy, you have your reds. So that's part of your R for Roy. Orange coming on through here, Y, and that's going to be getting faster and smaller waves. Transfer over to green, and then blue, indigo, and then violet will be the actual smallest waves that carry the most energy. So they don't go near as far as the red. Next, you come into your X-rays. So these have more energy, but don't travel as far. The energy in these could actually cause radioactive damage in our bodies and our tissues. So they suggest you not have too many X-rays, or if you're somewhere where they do X-rays, they have a specific area for you to have those X-rays in where it doesn't affect other people. And then you go to your gamma rays. These are going to be the strongest waves. They carry the most energy, they do the most damage, but they don't travel as far as the other waves. So don't travel as far, small waves, but have the most energy so they can do the most damage. All right, so with this visible light, it's going to make up a small portion, as you see, of that electromagnetic spectrum, so about 400 and 700 nanometers. This was determined by a Scottish mathematician named James C. Maxwell, and he discovered this in 1865. Quantum theory of light and photons is going to be proposed by a German physicist, Albert Einstein, in 1905. Now, most of us have heard of him. He said that visible light is going to be composed of small individual packets of energy known as photons. These photons are going to be coined by an American chemist, though, named Gilbert N. Lewis in 1926. So the idea came from Einstein, but the actual naming of these particles is going to come from Lewis. Characteristics of photons that Einstein was able to determine is the intensity or brightness of light is going to depend on the number of photons that are being absorbed per unit of time. With that, each photon is going to carry a fixed amount of energy that is determined by how that photon vibrates. The slower the vibration, the less energy that photon carries. So that would be your red waves, your yellow waves. The faster the vibration, the more energy that photon carries. So now you're getting over to your blue, indigo, and violet waves. So they have the most energy, but they are going to be the smallest waves. Now, photons travel, like we said, in waves, and so the distance a photon moves during its complete vibration will be referred to as its wavelength. So wavelength of visible light is going to be measured in nanometers or billionths of a meter. That's going to range from 400, where violet is, to 700, where red is, in those nanometers. And so notice that wavelength gets smaller at violet at 400. It gets larger or bigger waves at 700 at red. So photon energy is going to be inversely proportional to the wavelength of light. So the longer the wave, less energy per photon. Shorter the wave, the greater the amount of energy per photon. So three fates when photons strike an object. It could be reflected, that's where light bounces off of the object, and that determines the color of the object. So if you are wearing white, if all that light is being reflected off of it, that's every color makes white. So all that light being reflected off of it makes your shirt appear to be white. If your shirt has all the colors in it and it's absorbing, then it will be black. So white has no color, it's reflecting all light back, so you see white. Black has all color, so it's absorbing all that color, and it's reflecting nothing back, so it appears to be black. If you have on a green shirt, then your shirt actually has every color in it but green. It's reflecting green back, so you see green. If you have on a shirt that is red, it's reflecting every color, or it's absorbing every color, made of every color, but reflecting red back. So everything you see is a reflection. Actually, that's the color that is not contained in that object. That's the color that's being reflected back. So leaves are green because pigments they possess reflect green wavelengths. If you were to put a plant under green light, well, it's reflecting green light back. So it's not going to be able to use that light to photosynthesize. So that plant will die. White is going to be objects that reflect all wavelengths, but they don't have any of that color in them. So they absorb none. Black is going to be objects that reflect no light, but absorb all because they are every color. So reflection is one fate. Transmission or transmitted, that means the light can pass through an object and that determines the color of the object too. Or it could be absorbed, and that causes an effect and function of photosynthesis. So it's absorbing the light, and when it absorbs all that energy coming in, it then changes it over to another form. Photosynthetic pigments are going to be very, very important when plants need to be able to process and make food. So it's going to be found within the thylakoids of the chloroplast, and it's responsible for absorbing all the different wavelengths of light. So major photosynthetic pigments will be chlorophyll A, chlorophyll B, and carotenoids. Chlorophyll A is going to be your most abundant photosynthetic pigment. It's going to help to absorb violet, blue, orange, and red wavelengths of light. It's going to reflect and transmit things like green wavelengths, and it gives leaves their color. Chlorophyll B is going to absorb different blues and orange-red wavelengths, and it reflects and transmits greens and yellow wavelengths, which contribute to leaf color. So with this one, it's going to be there all the time, but as the light and temperatures change in the fall, you have less light, the chlorophyll A starts to die out, and some of the last pigment that you see is going to be the chlorophyll B. And so that's when you start to notice your different yellows, or with the chlorophyll A, it starts to die out and you'll start to notice the different colors basically. And the carotenoids are going to be the big ones. This is going to give you your yellows and your oranges and your beautiful reds that you're going to see in things like maple trees. And so chlorophyll A starts to die out, then chlorophyll B, and eventually the carotenoids are left, but then they start to die out. But those are going to give you your beautiful fall colors. All right, so with the carotenoids, they're composed of carotenes and xanthophylls. These absorb violet and blue-green wavelengths of light. They reflect yellow, orange, and red wavelengths. And so often they are masked, like I said, by chlorophyll A and B during the summer because there's so much of them. But as that weather changes and there's less sunlight to be used, the chlorophyll pigments start to break down a lot sooner, and you're left with those carotenoids that give you the beautiful autumn colors. So here is the chemical structure or makeup of chlorophyll A, B, and the carotenoids, and you can see that electromagnetic wavelength of colors, or your Roy G. Biv there. So photosynthesis is going to involve the conversion of carbon dioxide and water along with light energy into glucose and oxygen with the addition of that light energy. So the reactants are going to always be on this left-hand side. So these are your reactants: carbon dioxide, water, solar energy or light. What you're making, these are going to be your products. And so you're producing sugar, that's your glucose, C6H12O6, and then O2 is breathable energy. Breathable oxygen. All right, so light-dependent reactions. This is going to be the photo part of photosynthesis. So it's responsible for absorbing the solar energy and converting it into your ATP molecule and a energy carrying molecule, which is NADPH. So that's your high energy carrier. It stores the necessary chemical energy to help synthesize glucose. Oxygen gas is also going to be produced either released through that stomata, or it's going to be used by the plant during cellular respiration. So plants don't just need carbon dioxide, they also need oxygen. These reactions are going to occur in a specialized unit called photosystems, and those are going to be located within those thylakoids. Remember those little stacked pancakes that we saw inside the chloroplast. So there's two main regions: the light harvesting pigment complexes, which contain about 400 photosynthetic pigments which are going to be bound to proteins, and you have a reaction center. That's going to be surrounded by the light harvesting pigment complexes and contains special chlorophyll A molecules and proteins in there. So photosystem 1 or PS1 and then photosystem 2 is PS2 are going to be the names of these systems. They don't reflect the order in which they occur. That's actually the order in which they were discovered in. So actually photosystem 1 occurs after photosystem 2, but it was discovered first. So that's how they got their names. So several scientists are going to be responsible for the discovery and outlining these functions of the two photosystems. That's going to be Robert Emerson, William Arnold, Hans Gaffron, Kurt Wohl, Robert Hill, and Fay Bendel. So we have them to thank for these discoveries of photosystems. So these reactions are going to begin at the photosystem 2. Remember, it was discovered after photosystem 1, but it actually occurs first. And it's going to end in photosystem 1. So photosystem 2, solar energy is going to be absorbed by photosynthetic pigments in the light harvesting pigment complexes. It's going to pass from one pigment to another until the energy reaches the reaction center. Within the reaction center, you're going to have specialized chlorophyll A molecules that receive the energy and they excite the electrons that are there. And those are going to be donated by splitting of water molecules. So it takes water, excites them, and it splits that molecule, and you've got that electron. The remaining hydrogen protons result from splitting water molecules are going to be used to make ATP and NADPH. Remember that high energy molecule carrier. And then oxygen atoms are going to combine to form oxygen gas, or O2, that we breathe. Once they're released from photosystem 2, the high energy electrons are going to travel down what's known as the electron transport chain, where the energy is going to be lost to a pump, which is pumping hydrogen protons into the thylakoids, and that results in a proton gradient. That is going to increase the number of hydrogen protons in the thylakoids that travel through ATP synthase to make ATP. Now, synthase is going to be an enzyme, so the enzyme is going to help to create the ATP. Anytime you see ASE or ACE behind something, that's what it is, an enzyme. Remember, enzymes help to speed up reactions, increase the amount of or decrease the amount of energy required because it helps to produce that reaction. So now the reaction center of PS1, which is your second step, is going to receive those low energy electrons and the absorption of the solar energy by photosynthetic pigments, and that excites the electrons again. From there, you have high energy electrons that are going to leave that reaction in the PS1 center and travel down another electron transport chain to be transported or transferred into the NADP plus to form NADPH. So that's the carrier particle that's going to take it. So this is basically what you're seeing going on there. Now, the light independent or synthesis reaction is going to be the part that doesn't require light energy. So you have the photo part and you have the synthesis part. So this is the synthesis part. This is going to occur within that jelly-like substance, or kind of like the cytoplasm of the chloroplast. So it takes it from the thylakoid over to the stroma, or that gel. This is going to use the energy stored in ATP and NADPH formed during that light independent reaction and converts the carbon dioxide into glucose. So that involves a series of enzymes that are catalyzed reactions that occur at what's called the cyclic pathway, known as the Calvin cycle. So the Calvin cycle often is referred to as the light independent reaction. This is going to be named from the scientist Melvin Calvin, who, with his team, discovered a series of reactions that involved this process during the 1950s. It's going to be a three-step process. The first step, you have what's called carbon fixation. That involves attaching carbon dioxide to ribose, or ribose biphosphate, or Rub. And that's going to help to form the enzyme Rubisco. That helps to produce an unstable 6-carbon molecule, which is then going to split to form 2 3-carbon molecules known as 3PG. Second step, you have the synthesis of glycerolaldehyde 3-phosphate, or G3P. That begins with 3 G3P or 3 PGs, it is converted into a series of enzymes that's catalyzed in reactions using ATP, NADPH, and that's produced during that light reaction, and then it's going to take it and form it into the G3P. The G3P molecules are going to then be combined to form 6 carbon sugars, such as glucose, and an additional complex carbohydrate like starch or even cellulose that plant could use. Third step, you have regeneration of that ribulose biphosphate or RUB-BP, and that's going to basically take some of that G3P molecule, used to regenerate that, and then use energy from ATP. And so that's what's going on here. You've got your ATP energy coming in. You've got your ribisco biphosphate comes in with carbon dioxide, makes your 3-phosphoglycerate, bring in some energy, break off that third phosphate group. Then you have your G3Ps there. NADPH comes in, releases some of those high energy electrons, makes your G3P, and then it goes through the process again. Sometimes with photosynthesis, you need alternative pathways, and this is going to occur in times when you can't perform photosynthesis under normal conditions. So it's developed because of evolutionary adaptations to help conserve things like water in really dry climates or high heat or a lot of wind. C3 plants are going to be plants that are known as photorespiration. So these are going to be the ones that are able to close their stomata during hot, dry weather and reduce water loss due to evaporation. So closing the stomata helps to prevent carbon dioxide from entering the leaf, and it actually prevents that oxygen from exiting the leaf. So you're going to end up with a buildup of oxygen and a depletion of CO2. This is going to increase, like we said, those concentrations. And so that oxygen increase within that leaf begins to become fixed to that rubisco, and that's going to help to produce CO2. It does not produce ATP or glucose though, and so it decreases that plant's photosynthetic output. You'll see this in a lot of grasslands where you have things like wheat and corn being produced. The next one is going to be CAM. Oh, sorry, C4. I must have went too far. So the C4 is going to be, your corn. You have wheat in the other one. This is going to use a 2-stage carbon fixation process that helps an enzyme known as PEP. PEP carboxylase has a strong affinity for carbon dioxide when the concentrations are low. So during hot, dry weather, these C4 plants will partially close their stomata and allow the carbon dioxide to enter. So they don't close it all the way. Carbon dioxide entering the stomata is going to bind to those PEP carboxylases, and that's going to form a 4-carbon molecule such as malate. That's going to help or then be moved to parts of the leaf where the Calvin cycle can occur. With this additional step of fixing that carbon and using that PEP carboxylase, that's going to require more energy, which is kind of a disadvantage to the C4 plants, so it takes more energy to actually create the energy that they need. But it's an alternative process that is going to be needed for areas that could be hot or dry. This process is first reported in 1954, but it became extensively studied, and it was named by an Australian scientist in the mid to late 1960s. Now going to our last one, which is going to be our CAM photosynthesis, or our CAM plants. These plants are going to need to be able to conserve as much water as possible, and of course they do this by closing their stomata during the day when the weather's really hot and really dry, and opening it at night, allowing carbon dioxide to enter and the oxygen to leave. It's going to reduce the likelihood of photorespiration because the high concentrations of carbon dioxide are going to be collected at night, which is then going to bind to that PEP carboxylase enzyme, and during the day it forms malate. So that malate is going to enter the chloroplast. It releases the carbon dioxide when it does this, and that's going to be fixed through the Calvin cycle. Insufficient amounts of CO2 collected during the day are going to reduce that plant's growth. And in addition, it also reduces the energy required to be used by the PEP carboxylase. So it's considered a disadvantage to the CAM plants. This is why plants in these areas, like deserts and stuff, cacti, things like that, grow at a very, very slow rate. So this alternative process for them was first detailed in writing by a Swiss botanist, Nicolas Theodore de Saussure, in 1804, and by a French botanist, Ephraim Albert, in 1892. And then we have an American botanist who's going to also study this and describe it, Herbert Richards, in 1915. And so with this, the terms crossylation, acid metabolism, or CAM, is going to be introduced to our science talks, and that's going to come from Thomas in 1947. So with him, he's going to detail these studies that were performed in the late 1960s, and he's going to talk about, so that's where it finally gets its name from, in the 1960s. All right, so now we've made our energy. Now we need to be able to use the energy. And when we use this energy, it's going to be referred to as cellular respiration. So this is going to be how these cells are able to take the energy and use it to run the cell. Its net equation is going to be opposite photosynthesis. So it's the exact reverse of it. So we have, sugar coming in as a reactant, and we have oxygen that's going to be required. They're going to produce carbon dioxide, water, and ATP energy. All of this is going to occur at this organelle right here called the mitochondria. So what's happening? Aerobic metabolism, or aerobic metabolic process, is going to be where these cells extract energy from the complete breakdown of glucose to form what we said was carbon dioxide, water, and ATP molecules, or energy. This is going to occur, like we said, in that mitochondria. As these chemical bonds in glucose are going to be broken down, the molecule is then going to be rearranged into intermediate forms and release energy. The energy is then transferred by high-energy electron-carrying molecules along the electron transport chain, where it can make anywhere from 32 to 36 molecules of ATP per glucose molecule. So lots of energy molecules. The remaining electron is going to combine with oxygen and hydrogen atoms to form water molecules. This is going to be a very efficient process. It'll extract about 35 to 40% of available energy in glucose molecules, with the remaining energy lost as heat that's being performed by these mitochondria. So it occurs in 4 main steps. First thing you're going to have is glycolysis, then preparatory reactions, the Krebs cycle, and lastly, the electron transport chain, which is going to give you your most ATPs. So starting out with the first step, glycolysis. Hundreds of years ago of experiments and research that's going to be completed by Louis Pasteur, Edward Buchner, Arthur Harden, William Young, Gustav Embden, Otto Meyerhoff, and Jacob Parnas, is going to be outlined in our current understanding of this process. So these are going to be the scientists who help discover and go into depth with these processes. So the glycolytic process, as we know today, was going to be described by the scientists in the early 1910s that we see below. What it is it's going to be an anaerobic process that occurs inside the cytoplasm of the cell. Now with these, when you see them on your quizzes and on test questions, you might have one that says, okay, where does this occur? And you need to know the exact place. So where it's cellular respiration, first step glycolysis. Anaerobic means it does not require oxygen, and it's going to occur in the cytoplasm. So those are some things that you need to know. This is going to require 10 enzyme catalyzed reactions to break down, convert, and rearrange glucose molecules into 2 pyruvic acids. During this process, it's going to have to actually start it off by using 2 ATP molecules, so it requires energy to even start this process. So it begins with the 2 ATP molecules that activates the glucose molecule. Once you activate the glucose molecule, it's going to result in an unstable molecule, which allows these enzymes to begin to rearrange it. And it then produces those 3 carbon molecules called the G3Ps. So two G3P molecules begin losing their energy electrons and releasing energy as they proceed through the different chemical reactions. So as that molecule is going to be converted to 2,3 carbon molecules of pyruvate, it's losing some of its energy. So those high energy electrons that are going along with the high hydrogen atoms there are going to be added to two NAD pluses, which are coenzymes that form NADH. So you'll end up with two of those. And those are going to be responsible for transporting that high energy electron to the electron transport chain. So energy released during the breakdown of glucose is also going to be used to attach that phosphate to the ADP and make it ATP. So when it does this, during glycolysis, it will make 4 ATPs. But remember, 2 of those will have to go back into the next glycolysis reaction. So you're actually only getting 2 usable ATPs. It makes 4, but 2 have to go back and start the next glycolysis. So only 2 ATPs are kind of usable for this, for energy. So your overall net gain of ATP during glycolysis is 2 ATP, because 2 are going back to reactivate that glucose molecule and to start that glycolysis again for the next reaction. If oxygen is available, the 2 pyruvate molecules will then enter the remaining steps of cellular respiration. But if oxygen isn't available, the pyruvate molecules will be broken down within the cytoplasm during fermentation. So the final products of glycolysis are going to be 4 ATPs, 2 net, 2 pyruvate molecules, and then 2 NADHs. So what that means is 4 ATPs, but 2 have to go back. So 2 net's going to be the 2 that you can use. 2 pyruvates, and 2 NADHs, which are your high energy electron carriers. You go into the preparatory reaction, which is going to occur when oxygen is available. You're going to take the 2 pyruvates and enter the mitochondrial matrix. So we went from the cytoplasm to now going into the mitochondria. The matrix is going to be the gel that fills up that mitochondria. So while in the mitochondrial matrix, you're going to have a series of enzymes catalyzed into reactions to convert the pyruvate molecules into molecules that can then enter what's known as the Krebs cycle. So pyruvate molecules are going to react with coenzyme A, or CoA, and that's going to remove carbon and oxygen atoms from the carbon dioxide, so it breaks them apart. Additional modifications to the pyruvate molecule is then going to remove high energy electrons, which are going to then be added along the hydrogen atoms to NAD+, and that forms the NADH molecules. The remaining 2 carbon molecules is then going to attach to the CoA to form acetyl-CoA, and those molecules are then going to be carried to the Krebs cycle. So 2 pyruvate molecules enter the mitochondria. The final products are going to be, in that preparatory reaction, are going to be 2 molecules of acetyl-CoA, 2 molecules of carbon dioxide, and 2 molecules of NADH. Right. All right, so the Krebs cycle. It's named after Hans Krebs, although key components of the reaction or of this process are going to be discovered by other scientists that you see. So with the Krebs cycle, it occurs within the matrix of the mitochondria and involves 8 enzymes that are catalyzed reactions called the cyclic pathway. It begins when each acetyl-CoA molecule is going to form a preparatory reaction, and it combines with the oxaloacetate and 4 carbon molecules to form a 6 carbon molecule. And so that 6 carbon molecule is then going to be rearranged into different types of intermediate molecules. That rearrangement produces high energy electrons, which then are going to be transferred to NAD+, FAD, and ADP. When they do that, they form NADH, FADH2, and ATP. The NADH and FADH2 are going to be your high energy electron carriers that will transport electrons to the electron transport chain. Carbon dioxide is going to be produced when carbon and oxygen atoms are going to be removed from that 6 carbon molecule when it's rearranged. The remaining 4 carbon molecule is then going to proceed through a series of enzymes that are catalyzed reactions to produce the original 4 carbon molecule, the oxaloacetate, and that causes the cycle to repeat itself. So since you have 2 acetyl-CoA molecules that are being produced during the preparatory reaction, the Krebs cycle then turns twice. So it goes through this process twice to create what it needs. The final products at the end of the cycle is going to be 2 ATPs, 6 NADHs, 2 FADH2s, and then 4 carbon dioxide molecules. And so this is what's going to be released as a organism's breathing out. And so here is a visual, and it goes through this cycle twice. The last stage is going to hit the electron transport chain. This occurs along the membrane-bound proteins that are located within the mitochondria, cristae. So we went from glycolysis in the cytoplasm to the preparatory phase in the Krebs cycle, which we're going to be in the matrix of the mitochondria, the gel-like or cytoplasmic-like substance, to now we're actually moving to the membrane-bound proteins and what's called the cristae. So these proteins are going to act as high-energy electron carriers like NADH and FADH. They're going to pass these electrons down the electron chain, release the electrons, so now these molecules become NAD plus and FAD, and they return to the Krebs cycle to be reused again. Now, the energy that was released from these molecules is going to help pump hydrogen ions across the mitochondrial membrane and produce what's known as an ion gradient. So, as hydrogen ions pass back across the mitochondrial membrane, they flow through ATP synthesis, an enzyme that uses energy to form the ion gradient to produce ATP. So this process is often referred to as chemiosmosis, and it was first proposed by Peter Mitchell in 1961. Upon reaching the end of this electron transport chain, electrons are going to combine with oxygen, the final electron acceptor, and hydrogen atoms are going to form water molecules. And so here you see all of that happening. Moving on, what do we get from this? So ATP yield can be variable. A completion of cellular respiration is going to result in anywhere from 32 to 36 molecules of ATP produced from every glucose molecule that was broken down. The actual yield of ATP molecules produced will vary for different reasons. You could have NADH and FADH2 molecules that may not always be used to generate ATP. Studies have shown that 2.5 molecules of ATP are going to be generated for each NADH molecule and 1.5 for every FADH2. So that would equate to about 25 molecules of ATP from NADH and about 3 molecules of ATP from the FADH. And that gives you about 28 ATP molecules. Addition of that 4 ATP that you got from glycolysis and the Krebs cycle, so you got 2 from each, and you combine that to make 32 ATP. So on average around 32, but can be 36. Respiration rates in cells may produce a variable ATP. Muscle cells are going to be highly active and require more ATP, while adipose tissue is going to be less active and require less ATP. So the more muscular you are, the more energy you need. Not all hydrogen ions are going to be transported across the mitochondrial membrane, and so there you have theoretical number of ATP molecules produced. It's going to assume all hydrogen ions are going to be transported. So if some aren't, then the ATP is going to be less. Intermediate forms during Krebs cycle are also going to be used to make proteins and fats sometimes, so not necessarily ATP molecules. Fermentation is going to be a metabolic process that's going to be used for thousands of years to produce things like alcoholic beverages, or can be used to produce different types of foods. Think of things like yeast breads and pizza crusts. Experiments are going to disprove that air caused fermentation, and those experiments began in the 1930s. Theodor Schwann in 1936 concluded that fermentation was a result of yeast, which is going to be a living organism. And so he's going to be the one on the left. Louis Pasteur in 1856 is going to demonstrate that yeast are going to be reproducing and growing with or without the presence of oxygen, and they produce alcohol during fermentation, while bacteria are going to produce acids. So this is going to lead to a process that we know as pasteurization. And so Louis Pasteur is going to be the picture in the middle. Edward Buckner in 1897 is going to conclude that enzymes produced by yeast cells are going to be responsible for chemical reactions that are associated with fermentation. And so he's going to be your image on the right. So fermentation is an anaerobic process. That means it does not require oxygen. So it occurs in the cytoplasm where carbohydrates are going to be broken down and use different molecules as their final electron acceptors. So during glycolysis, the glucose molecule is going to be split to those two pyruvates, but without oxygen, those electrons are then going to be transferred from NADH to pyruvate molecules. So two different types of fermentation, depending on which molecule is going to be its final electron acceptor. So you can have what's called lactic acid fermentation, and this one I like to refer as the one that your muscles make when you're exercising and your muscles start to burn. Usually during those times, you're breathing pretty hard because you are running out of oxygen that your body could use for metabolism. So with this, it's going to occur when pyruvate molecules and their final electron acceptors are going to be converted over into lactic acid. That's like I said, a common process in muscles when oxygen levels are going to be low and pyruvate molecules are going to exceed oxygen delivery. And so those cells are being vigorously worked, and so they start to build up as lactic acid there. That lactic acid is going to bind to those muscles and cause muscle fatigue, and that's what actually helps you, like you're feeling the burn. This is what's causing that burn. Acid's kind of grimy, so as those muscle cells are extending and contracting and coming back and going back and forth with that acid there, it causes a burning sensation or a heating up of that muscle, which causes muscle fatigue. When oxygen levels return to normal, the lactic acid's then going to be delivered to the liver, and then the liver is going to convert it back over to pyruvate, and it can enter cellular respiration from there. It's going to be an important process in making food such as cheeses, yogurts, and chocolate. The second type is going to be alcohol fermentation. It has a two-step process where the pyruvate is going to be converted over to acetaldehyde. That's going to act as the final electron acceptor and result in the production of ethanol. Important process is in making alcoholic beverages such as beer and wine and foods like breads. So your yeast rolls, and especially like certain types of bread doughs that are going to be used in pizzas. Advantages of this process aren't only going to be for humans in the production of food and alcoholic beverages, but for bacteria and archaea as well, because this enables them to produce ATP under anaerobic conditions, so when they don't have oxygen. Disadvantage includes it's an insufficient energy extraction. You actually get about 2% of the energy, and it produces toxins that can be harmful to the cell. So acids produced during lactic acid fermentation will lower the pH of the cell's environment, and it affects how the enzymes inside that cell work, so they might not work efficiently. Alcohols produced during alcohol fermentation will cause proteins to denature, and that affects their overall function. Remember, denaturing or denaturation causes proteins to change their shape, and change the shape, you change the function, and it can make it non-functional. That's going to end us with our energy, photosynthesis, and cellular respiration unit. I'll see you back for the next unit.